Without further ado, I want to introduce our first speaker, who is a friend of mine who spoke in a workshop with Deepak the other night. He is Rudolph Tanzi, PhD, the Joseph P. and Rose F. Kennedy Professor of Neurology, Vice Chair, Department of Neurology, Genetics and Aging Research Unit, Massachusetts General Hospital, Harvard Medical School, and a musician who, as I mentioned the other night, jams with Aerosmith and has a new album coming out with Joe Perry, Johnny Depp, and Iggy Pop, if I'm not mistaken, maybe a few others. So he's a well-rounded guy, to say the least. Please welcome Rudy Tanzi.
I think that's the first time I've ever been introduced for a talk with the words Iggy Pop in the introduction. So, yeah, that's a good one. Anyway, good morning to all of you, and thank you for being here, early in the morning for the first talk. I want to also thank those of you who, and I'm not sure if any of you are here, who were there last night at the party, singing at the piano and still got up to come in. We had a great sing-along last night. And I know that when I was speaking with Deepak the other night, I said, religiously, I get 7 to 8 hours of sleep. Well, I broke my religion, and it didn't get enough, went to bed a little too late. So the goal of my talk today, two goals. It's about Alzheimer's disease, which is, probably the biggest punch in the face consciousness can get in terms of, keeping your mind and keeping your brain intact as you get older. And the two goals are, one, to talk about what does Alzheimer's do to you in terms of what does it do to the brain? What does it do to the mind and consciousness? What does it do to the patient? What does it teach us about self, given what happens to a patient during the course of this insidious disease? And secondly, I want to spend time telling you that there's a lot of hope. And, you hear a lot of things about Alzheimer's in the news, like popcorn cures it and aftershave and things like this. And especially, it has to be a particular, I think it's Old Spice that works. And, but the thing is, there's a lot of misinformation. So my hope is also, at the end of this, that you will have the newest state-of-the-art understanding of what causes this disease and how we're going to stop it. And I got to tell you, I'm very optimistic that over the next 5 to 10 years, if we have enough funding to get it done, that we will stop it. And that I think for younger generations right now, that hopefully they're not going to have to worry about this disease. They'll talk about it in the past. And so just a quick introduction, Alzheimer's itself is the most common form of dementia in the elderly. Many people ask the difference between dementia and Alzheimer's. Well, most dementia in the elderly is Alzheimer's. And we say there are 5 million patients in this country, 50 million worldwide, which is a lot. But as I'll show you, we don't say you have Alzheimer's until you have symptoms. And in contrast, in cancer and in heart disease, we say you have cancer when you have a tumor without symptoms. We say you have heart disease as soon as there's plaque and calcification and you're treating it, but you haven't had symptoms yet. If you take the people who are on their way to Alzheimer's disease based on pathology, who don't have symptoms yet, that 5.4 million number goes up to about 20 to 25 million, making it perhaps the most prevalent disease in this country. And I think the paradigm shift is coming where now we can define the disease as pre-symptomatic and symptomatic Alzheimer's, which will increase the numbers, and I'll explain more about that later. And this pathology begins in virtually everyone, especially after 40 years old. It just comes with age, and the question is not, whether you're going to get this disease, but when. It's with few exceptions, a few lucky mutants out there. You basically, if we live to 120 years old, everyone would be at least in the earliest stages of symptoms of this disease. And right now, if, that number's a little outdated. For people over age 85, it's about 30 to 40% have the symptoms of AD, many more with the pathology. And given the current lifespan is 80 years old, think about what's going to happen as we keep lifting the lifespan from 80 to 85 with that type of number. It's going to just wipe out Medicare and Medicaid. As you might imagine, the number one risk factor is age, then family history. Women get it more than men. 2/3 of patients are women, and it's not clear why. I'll talk about that a bit later. Head injury is a risk factor, stroke and neurovascular factors, and even emotional trauma being emotionally traumatized, and getting that big cortisol hit can be a risk factor. And the current drugs out there, Aricept and Namenda, they're in that category of better than nothing, but just about, barely better than nothing. They treat the symptoms, modest effects and temporary effects. So we got to do a lot better than that. So a lot of people ask me the difference between Alzheimer's and just getting old, having senior moments. And I illustrate that here, caution, senior moment in progress. This is my favorite because I do this all the time. I know I came into this room for a reason. I do that all the time. I go into the kitchen, my cat follows me, we both just stare at each other, we have no idea why we're there. And then I like this one, the two dinosaurs looking at the ark, kind of floating away, saying, oh crap, was that today? Just losing, not pain, losing track. And the thing is, when is this Alzheimer's, and when is it just, getting older and changes that come with getting older? And I use this as an example, like probably if you're old enough, the day JFK got shot, or when the shuttle exploded, or 9/11 more recently, like how many of you remember in utter detail everything you were doing on 9/11, during and after the attacks? Raise your hand. So that's like almost everybody. See, because what happens is when there's a lot of emotion attached to events, then you really consolidate memories well. You register memories, you solidify those memories. And the thing is that this is what we do less of when we get older. And so kids do this all the time. I'm a late life parent. My wife and I had our first kid in our 40s, and my little girl just turned 8. And for her, that's a boy. That's not my little girl. But for her, every moment's like a wow moment. Like everything's so cool, everything's so amazing, right? And then you get older and you become this guy. And say, so what? Oh yeah, I heard that talk last year. Same thing. Oh yeah, there's one extra slide. So what happens is we get jaded, we get apathetic, we have less passion, we don't live in the moment. And the question is, how much is it just that we're not living in the moment, being present, being thrilled with the world around us, and thus learning better, because you learn better when emotion's attached? Or is it that pathology, is it Alzheimer's pathology that's making this guy a little depressed, a little less interested, a little more jaded, a little more apathetic? So we'll get a little bit into that. Now, in Alzheimer's itself, this is a catastrophic cognitive decline. This means you at some point you can't make sentences, you can't follow complex tasks like making a complex recipe, eventually you lose track of time and space. And then the early signs of the disease is agitation, there's depression. And this is different than just, forgetting a name or losing track of where you left your keys. But when is it early Alzheimer's pre-symptomatically, and when is it actual Alzheimer's disease? And I think the only way we're going to really know that, and we can do that, as I'll show you, is we have to basically diagnose this disease before symptoms hit. And you have to treat this disease, not prevent it. We say prevent disease, but you have to treat this disease before there are symptoms, just like we do with cancer. And there's a tumor. Remember, people used to be ashamed of cancer, and they had the C word. Well, it's going to be the same thing here. You're going to be embarrassed that you already have Alzheimer's pathology at 50 or 60 years old. It's the A word. But the future is going to be, don't worry about it. If there's pathology going, we're going to stop it before there are any symptoms. That's how we're going to stop the disease. So that's just a preamble to what I'm going to show you. So this is, if I can get this to work, this is a slide of a new method called Clarity, where you can see into the brain by stripping the fats. This is a mouse brain. And I show this because this is the hippocampus of a mouse. The hippocampus is what's affected in Alzheimer's disease. This is where you have short-term memory. It's your limbic system. It's where you live. It's where you have fear, you have desire, you're keeping track of information during the day, you're storing short-term memories, and you're seeing the neurons here and synapses. And this is basically, even though it's a pretty small area, hippocampus means seahorse in Greek, and it looks like that. And it's right, if you went right through here. And this is the area that gets decimated first in Alzheimer's disease. So, overall, your brain contains 100 billion neurons, hundreds of trillions of synapses, or up to a quadrillion. And these are allowing you to access all these memories of the experiences that define your life. And over time, as I'll show you, Alzheimer's just destroys this neural network. So it's like you built this big tapestry of synapses based on your experiences, and then Alzheimer's just rips it apart thread by thread, and it gets faster and faster as it goes on. It's just a terrible disease. And this is Dr. Alzheimer here. This is his patient, August Dieter, and she was a 57-year-old woman who was admitted to an asylum in Bavaria because her husband said she was getting insanely jealous. So it's kind of weird back then. You got thrown in an asylum because your husband says you're jealous. But this woman was having big problems. And he said this was when she was 55. She died at 57. She's sitting on the bed with a helpless expression. This is from Dr. Alzheimer's journal. And he interviewed her. He said, "What is your name?" And by the way, see this circle? This is the old man circle. You know, I can never find my little arrow, my cursor. So I downloaded this app for $2.99, and I can put a circle around my cursor. But I highly recommend it if you can't find your cursor. Just looks a little funny. So anyway, Alzheimer asked her, "What is your name?" And she said, "August." That's good. Last name? August. Well, that's not so good. What's your husband's name? August, I think. So she's stuck on that. How long have you been here? And she said 3 weeks. She had actually been there just 2 days. And then he said that she broke down and cried hysterically and just kept telling the doctor, "I've lost myself. I've lost myself." And that's what this disease does, is that you're there, but there's this fog in your brain, and you cannot access yourself. And that begs the question, what is self when you can't access it? What's happening in Alzheimer's disease that says you can't access self? And I want to say that, this is the self that's, I guess, working with Deepak, this is the false self. This is the ego self. This is the self that's been created by lifetime. But it's also the person that people know and love, right? I think the true self, the non-local self, it's always in there, of course. That's the true identity. We're talking here about the self that people know, that lives in the world, and is the culmination of all their experiences in life. And this is what's ripped apart. So how does that happen? So here we see the hippocampal area, short-term area, and then there's another area of the brain called the entorhinal cortex, which is red. It's just behind the nose. And basically, every day, sensory information is coming in. You're hearing things, seeing things, and that information is bundled into a big cable called the perforant pathway, because it perforates the hippocampus, and it tells the hippocampus, here's what's happening right now. And this is where short-term memory comes from. And then short-term memory, eventually, with repetition, becomes consolidated as long-term memory in other parts of the brain. But this is kind of the thumb drive before you download it onto the hard drive, which would be the frontal cortex. So in Alzheimer's, what happens is that the synapses in the hippocampus, where that perforant pathway is coming in, trying to tell you what's going on and make sure you remembered what happened a few minutes ago, it's those synapses that start to pull back and die. The actual spines on the neurons start to die. And so the big question became, hey, so what's happening here? Is it that when you're experiencing your day, is that information never getting stored? In other words, the cable sends the information to the hippocampus, but it just kind of fritters away. You don't store it at all. Or is that information actually getting stored, but you can't access it? And now there was an experiment that was just published in Science or Nature about 2 weeks ago from Susumu Tanagawa, Nobel Prize winner. And he answered the question in a study with mice. So I want to ask, how many people, just for fun, because I was wrong about this my whole life, how many people think that the information never gets stored when there's this problem with the hippocampal connections? Raise your hand. Never gets stored. Raise your hand. Okay, how many people think that the information's there, but you just can't access it? Raise your hand. All right, so you guys are a lot smarter than me. Because what's that? Oh, you read the paper. I forgot about the paper being out. Yeah. But for 2 decades, I gave this talk saying the information is not getting stored because my colleague Brad Hyman, that's what he published in 1986. And it turns out if you take mice and they have Alzheimer's mice and you teach them a task and the Alzheimer's mice can't remember it, right? And then optogenetically, they can turn those neurons on even though they can't get activated by themselves. So if you activate those neurons in the molecular layer, the dentate gyrus, where you're getting this information, they're just fine, they remember. It's just that the neurons can't activate because the spines are being removed and degenerating. So that's kind of good news. It means the information's in there, we just got to learn how to turn it on, right? So I thought that was great. So now what happens is, as your short-term memory goes, this eliminates the continuity in consciousness, because now new information coming in cannot be placed in context with your long-term memories. And a lot of the sense of self requires that as you're experiencing the world and learning things, you're associating it with what you already know. And so if information's coming in, but you can't access it 5 seconds later and place yourself in context of time and space, then this is basically leads eventually to a loss of self-awareness. And in this disease, what really happens in patients is they have a loss of self-awareness. They're living in the moment, basically. Eventually the late-stage patients are living purely in the moment. Information's coming in. They can't put it into context in terms of what they already know. And so it sounds great, living in the moment, but if you're living in the moment and you don't have self-awareness and when you don't have any self to associate it with, it's actually pretty horrifying because you just cannot there's no meaning to what's happening to you during the day. You're losing that context of meaning because you can't associate new things with what you already know. And so to a certain extent, it means that, when we talk about, there we go. When we talk about what's happening in Alzheimer's, the first thing that happens, actually this little circle is very handy, the first thing that happens is the limbic system's affected. So if you take, the triune brain model, brainstem, limbic system, neocortex, and use Dan Siegel's handy brain, where you tuck your thumb in, fold your fingers over, brainstem down here, limbic system tucked here, frontal cortex in the front, and the brainstem, of course, being the oldest, about 300 or 400 million years old. This is the instinctive self. No acquired memories here, all innate memories, somehow miraculously programmed in our genes. This is just fight or flight, find food, procreate, keep the species going. More or less of a selfish type of survival-based existence. The limbic system, you have the first essence of memories, and the first memories that occurred in the brain had to do with remembering pain, and that leads to fear, and remembering pleasure, and that leads to desire. I want it again. And then this is also short-term memory associated with these memories of it's good for me, it's not good for me, fear, desire. And then eventually, setting the map of reality for yourself, these memories get stored into long-term memories in the neocortex, the intellectual, rational self, also the area where you get meaning, you get purpose, creativity, and acquired memory. So in Alzheimer's, the first area attacked is the limbic system. So you're losing that everyday self where you live, keeping track of things. And then you resort to your long-term memory. So, the patient's talking about World War II or the day they got married in the 30s or 40s. And then eventually the disease spreads lastly to the brainstem. So that brainstem stays pretty much intact almost the entire disease. In fact, in many cases, it's not affected at all. And that's the course of the disease. Now, interestingly, the music memory area of the brain is completely spared in Alzheimer's disease. It's tucked away in the middle, and as the pathology spreads throughout the brain, music memory is not affected. So you see often in a nursing home an older person who's late-stage Alzheimer's who can sit at the piano and play songs and sing and remember all the words, because remembering words to songs is not in the same area as language assembly. It's a different area of the brain. So we just launched a little app called Spark Memories Radio, where we basically provide a program for loved caregivers where they put in the birthday of the patient and anything you know about their genre of music preference, and then it plays for them music that was popular between the age of when they were 12 and 25. Because it's between 12 and 25, going through puberty, high school, college, that you're really bonding emotionally with your music. And it's what really stimulates you. And then after 25, you still listen to music, you still say, hey, I like this band, hey, I heard this band, they were okay. But that music that really stimulates you was between 12 and 25, and that's what the program does. We're getting amazing emails back from people saying that they have a loved one with early onset Alzheimer's, and now they're not as depressed or agitated, they're calmed down. And even ones who have loved ones who are vegetative and haven't spoken for 6 months, they're saying they're tapping their feet, they're singing along, they're suddenly telling stories about a girlfriend or a car they had when that song came out. So we're hoping this will do some good. Now, let me talk a little bit about what the pathology is. And basically, amyloid deposits are these big gunky plaques shown here that deposit, outside of the neurons. And then you see tangles inside of neurons shown here. This is where the microtubule protein tau falls off of microtubules. Microtubules that are stabilized, the neurons die, and these tangles spread. If you follow the blue here, this is how the tangles spread through the brain, around and around. And so that's, and then the third pillar is neuroinflammation. And it turns out, and this is probably one of the most important things we learned about the disease in the last 5 years, is that once in a while you see resilient brains. These are people who die in their 80s and 90s, no cognitive problems when they died, perfectly fine. And then when the pathology is done, they happen to have an autopsy, you see their brain is full of plaques and tangles. So many plaques and tangles, the pathologist said, this is an Alzheimer's case. Nope, he was perfectly fine. And what you'll see each time is that there's no neuroinflammation. So it turns out, more good news, we can live with a head full of plaques and tangles, the main pathology of this disease, and never get symptoms if there's no neuroinflammation. So this is a very big point, and so I'll show you, in the next slide, these are the different genes my lab and other labs have found over the last 30 years, and we've grouped them into ones that affect amyloid pathology, ones that affect tangles and microtubules, and ones that affect this growing number of ones that affect inflammation or the brain's innate immune system. And it's true that, when tangles form, and especially when microtubules become destabilized, you start to have cognitive issues. In fact, along with Stewart's hypothesis on ORCA, I think that basically a lot of what's happening in people who are beginning to, some cognitive issues with age is that you don't have full-blown Alzheimer's yet, but because microtubules are destabilized, tangles are forming, that cognition is being affected. But then the catastrophic decline into dementia requires a kind of an auto-innate immune response to those tangles and plaques, and as neurons are dying, and inflammation steps in, and this inflammation that then throws you off the cliff. And my lab recently discovered a gene called CD33, which is the gene that is the on-off switch for inflammation. And it turns out that there are mutations in this gene. Some of them allow you to have lots of plaques and tangles and your brain stays chilled out and there's no inflammation. Other mutations allow your brain to overreact. So even with just a small number of plaques and tangles, inflammation ramps up. So a lot of the challenge going forward is to find one's inflammation set point. At what point will their brain react to the plaques and tangles, and then in this auto-innate immune response, kill many, many more nerve cells than those original plaques and tangles? That's a very hot area in the field right now. So in the first 4 genes, and I was involved with discovering the 3 first genes shown here. These are the early onset genes. They have mutations that guarantee early onset Alzheimer's disease. And then the late onset gene was ApoE. All those first 4 genes we found in the 80s and 90s all told us that the amyloid beta protein, this little protein that makes up the senile plaques, is the key, because every one of them somehow affected amyloid beta protein production or aggregation or clearance from the brain. It was all about how much amyloid you have in the brain. And then there were 3 decades of raging debate, because when you put the Alzheimer's genes into mice, they get amyloid, they eventually get some inflammation because of response to the amyloid, and if they live long enough, the mice get some memory impairments, but they don't get tangles. And people said, well, it's the tangles that are killing the neurons first, causing that cognitive problems, the microtubules are being affected. But in the mice, when you put in these amyloid genes, you don't get the chain of events, you don't get tangles. So how can amyloid be the cause of the disease? So there was a lot of debate about the amyloid hypothesis. So I have a member of my lab, Duyan Kim, he's shown here, and that's me. I call this my Green Hornet and Cato slide. And this was in the New York Times when they covered this thing we came up with where Duyan Kim, genius of a guy, said, look, 2 things. 1, humans are not 150-pound mice, newsflash. And 2, when people do try to grow neurons in petri dishes and try to get models using human neurons and cells rather than mouse cells, well, they grow them in media, right? That's liquid. And the second newsflash was the brain's not made of liquid, right? Except for that one scene in Brazil, remember the movie Brazil, where the woman's brain, it's a gross scene, but it haunts me every day. But anyway, the brain, if the brain didn't have a skull around it, it'd be like 3 pounds of Jell-O that would quiver. The brain's made of gel. So what he did was he made a system where you take human stem cells and we turn them into neurons and we put in the Alzheimer's genes and we sort them by FACS, and then we either grow them in a thin slice of gel, we call that the thin culture. And this would be like a thin crust pizza, basically. I didn't have breakfast. And then, or you can grow them in a thicker gel, which we call the deep dish pizza. And what that allows you to do is now you can grow these neurons, and they're living in a gel the way they would in the brain. And lo and behold, what happens is when you look at the gene expression, that you see all these gene expression changes, you actually see epigenetic changes where now the neurons, because they're in gel, right? I mean, picture trying to study humans and you throw them in a swimming pool, right? You're not going to get a good idea of human behavior if everything has to be done with them in a swimming pool 10 feet deep, right? So now these neurons are living in gel. They're like, yes, finally. And their gene expression is now matching what happens in the brain at every level. And it's matching much more closely to adult brain than what you see in a mouse brain. And what will happen is suddenly, see this orange dot? That's a plaque that forms in the dish in 6 weeks. And then if you wait just 2 more weeks, you get a tangle. This is called a paired helical filament. And what we could see was that the tangles follow the plaques, if you use this model. And if you use a drug like a drug I'm putting into clinical trials this year called a gamma secretase modulator that blocks the amyloid, see it's got rid of the plaques, then you don't see tangles. This was the first proof of concept ever that says amyloid causes tangles. The amyloid hypothesis is correct, you just can't do it in mice. And if you stop the plaques, you stop the tangles. So this kind of got the field back on its feet. The debate was over. Everyone accepted these data that tangles follow plaques, but that's still not enough to give you the disease. You still need the inflammation. So now we're using this to screen for drugs, and not just drugs, but non-invasive therapies. We're using radiofrequency stimulation, thanks to a postdoc that Anirban sent us, Subrata Ghosh. We're using direct current. And we're using classical drugs. And what we can do is we can screen neurons while they fire, high-content screening. So these are regular neurons 3 weeks into the dish, and I don't know what they're talking about, but they're happy. They're talking. It's like that party last night. Maybe they're singing. You can argue, is that consciousness? You know, a little piece of gel with neurons speaking to each other? And that's a 3-week culture. Now, this is 3 weeks, and these are neurons where we put the Alzheimer's gene in. Party's not so good. And you see these big blobs, they're loading up with calcium because they're becoming leaky. The membranes are starting to break down after just 3 weeks. 7 weeks in, the Alzheimer neurons. You know, not a whole lot going on, lots of blobs where there's leakage of calcium. So now we're screening both drugs and non-invasive methods to fix this, where we're actually screening against the activity of the neurons. So in case you don't believe that pathology matters, this says, let's see if we can bring these neurons back firing and interacting the way they're supposed to. And this is, and doing this, we already screened all of the US pharmacopoeia. So 1,200 drugs that are already approved for other purposes. We found 39 drugs that stop either amyloid and thus tangles, or stop tangles despite the fact that amyloid is there. And these are drugs that are already approved and used for other purposes, drugs for acne and asthma and things like this. So now we're thinking, can we take some of the safest ones, think about combinations, and start clinical trials with drugs you would have never thought would have worked. We couldn't do this when we just had mice. It was too expensive. But now we can screen, we can put these mini brains in little wells, and we can screen thousands of drugs at a time this way, and come up with answers 10 times faster, 10 times cheaper. So this was a big breakthrough for us and for the field. So that's Alzheimer's in a dish, as we call it. Now, I just told you the amyloid hypothesis works, but you can ask, for those of you who follow the literature, why, over the past decade, has every Alzheimer's trial targeting amyloid failed? Every drug, whether it's turning the amyloid off or unclogging the drain, getting the amyloid out of the brain, every trial has failed. Okay, so why? And this is where we have to go to Yogi Berra. Because Yogi Berra told us, he passed away last year, he said, you can observe a lot by watching. And so that's what we had to do. So now we can image for amyloid in the brain. We can see amyloid in the brain anytime we want with imaging techniques. And what we found was that the amyloid plaques form 15 years before dementia. 15 years before dementia, the plaques are accumulating in the brain. And if you track, and Yogi Berra said, we made too many wrong mistakes. Okay? And I'm a Red Sox fan. It kills me to show a New York Yankees emblem, but I got to show Yogi. His name's Yogi. So he said we made too many wrong mistakes. So look at this. This is amyloid, this red line here, going up. Mild cognitive impairment and dementia don't begin until here. The amyloid's done. It's already peaked. It's plateaued. The amyloid was going up here. What were we doing in these trials? We were treating people for amyloid here when they already had full-blown dementia, and the amyloid already did its job. And then you see the tangles going up over here. Inflammation's happening the whole time, but basically it was like if someone had a heart attack or congestive heart failure, went to the cardiologist, and you said, here, just take a Lipitor, right? You had to control, manage your cholesterol, diet, exercise 15 years before symptoms. That's what we were doing wrong. So now, as Yogi would say, the future ain't what it used to be, because what we're doing is trials, and these are some of the trials going on where we're treating patients very early in the disease, or even imaging people's brains, people who are at risk, seeing they have high amyloid, pre-symptomatic Alzheimer's, and saying we need to start treating you now, and seeing if we can prevent the conversion from pre-symptomatic to symptomatic Alzheimer's. These are some of the drugs that we're waiting to hear about. This is the drug I'm working on, allosteric gamma secretase modulators. I've been developing this drug for about 17 years in my lab. We're just finally going into trials. And so we'll see. We're going to start getting readouts next year into the next 3 or 4 years, but at least we're treating the patients, because it's not just right patient, right drug, it's right patient, right drug, right time, right stage of disease. Now, I just want to end by saying, even though we're targeting amyloid, we don't want to wipe it out, because we have a paper in Press in Science Translational Medicine coming out in May that basically shows amyloid is not just junk. It plays a role in the brain. It's an antimicrobial peptide. These are this is the defense system of the brain and the body, where you have these little peptides that actually fight bacteria, fungus, viruses, even cancerous cells. Amyloid, it turns out, is one of them. I don't have time to show all the data, except these are just graphs showing that in every case, flies, C. elegans, and mice, if amyloid is present, it protects against infection. It protects against infection from bacteria, viruses, and yeast. And what it does is the amyloid actually binds to a virus or bacteria in the brain, stops it from adhering to the cell, agglutinates it, and then the amyloid plaque is a big jail. It's a big trap. And if you can see down here, what we could see is if you have bacteria in the mouse brain, in the center of every plaque is one bacterium, where the plaque's trying to protect the mouse. So now what we believe is that with age, and we're seeing evidence now that where you're seeing yeast in Alzheimer's brain, we're seeing Borrelia, we're seeing viruses like herpes virus, small amounts of clinical pathogens, not enough to give you a meningitis, are now enough to trigger amyloid that's trying to protect you, and the amyloid deposits to do that. Once the amyloid deposits, that triggers the tangles, neurons start dying, there's cognitive issues with microtubules, inflammation, and death. So antagonistic pleiotrophy, something meant to help you, in the end, works against you, and we think this is the new model for the disease. So what can you do about it in the last few minutes? Physical exercise is associated with 60% decrease in Alzheimer's, at least 10,000 steps per day. 8,000 is probably okay if you have like a Fitbit or a watch. Mediterranean diet, stay away from red meat. Stay socially engaged, don't hole up at home. Ashwagandha and cat's claw, both are very good. Ashwagandha has been shown to get amyloid out of the brain. This is some data on a cat's claw extract I'm working on called Cognitive Clarity. It's a concentrated cat's claw extract from a vine in Peru. Wiped out plaques in mice, wiped out, I mean, it actually blows tangles into pieces. Very, very powerful, but you need a concentrated extract. And also learn new things. Just coming to this meeting, you're protecting yourself against Alzheimer's disease. You're learning new things, not brain games, but actually learning stuff. Meditation, I'll talk about, Deepak mentioned this in our panel, that we did a meditation study. I'm running out of time, so I'm speeding up. So basically, this was looking at the effects of meditation in a controlled clinical trial, and this was with Deepak running his Seduction of Spirit. The control group were just those at the resort. And we could see, as Deepak mentioned the other night, 40% increase in telomerase activity. This is what keeps cells young. Elizabeth Blackburn did this study. And then if we look at different gene expression, we can see that inflammatory genes, wound healing genes, viral infection genes, all are down. Basically, the body's chilled out. All of these genes that are compensatory to defend you, which can hurt you later on, like inflammation, are chilled out because the body feels that everything's okay. And also genes involved with amyloid accumulation in the brain, down. Amyloid levels by biomarkers in the blood say amyloid's down in the brain. This is 1 week of meditation, 1 week. So this is so this paper hopefully will be out soon. I call it compensatory gene expression chill out. That's what you get from meditation. That's my official scientific term for that. And then finally, sleep. And you know, I do try to get 7 to 8 hours of sleep. I didn't last night. thanks to the piano. But basically, during deep sleep, this is when your brain cleans out amyloid. The only time you don't make amyloid is during delta slow wave deep sleep. And it's the only time your brain actually clears it out. I call it mental floss. And it's also during deep slow wave sleep that those short-term memories in the hippocampus get downloaded onto the hard drive, the frontal cortex. So deep sleep's very important, and you really need to try to get 7 to 8 hours. And I'd like to say that if you're over 40 or 50 and you're not getting 7 to 8 hours of sleep, it's like that's the new smoking. You really have to get this sleep to clean your brain. It's not an option anymore, it's an obligation. If you can't sleep, you got to figure out how to get it done, go to a sleep clinic. But Deepak was talking about some melatonin ideas for low-dose melatonin, but it's very important to try to get sleep. And another way to get to do this is the Dreamweaver, and Dave Major is here, and Deepak and Dave Major and I came up with this device that uses light and sound to entrain the brain to get you down to that theta or delta state using different programs. And you can try one of these. There's a booth back where all the tables are, if you want to give it a try. I wrote the music and recorded it, and then David and his team brought in the light and the entrainment. And this delta one, the one that gets you down to a delta state, I think it's called Being Still. Actually, my wife has trouble sleeping, and it gets her to fall asleep. Of course, you have to get the glasses off. We have to work on something with that. But it is a great way to relax and get down to sleep. So, this just gets back to the two books Deepak and I did, Super Brain, Super Genes, where these are the big habits that you have to think about: diet, exercise, sleep, stress management, emotional stability, meditation. These are not only good for your epigenetics, your neuroplasticity, your brain, but probably the best way to stave off Alzheimer's disease, given everything that we're learning. And I wanted to end by saying the wisdom of the cell. We talk about this in Super Genes. If we were like cells, we'd be perfect, right? Cells with free will. Cells already automatically know what's good for it, what's bad for it, and what to avoid. They're totally living in the moment. They're focused. They're monitoring the well-being of other cells and serving cells around them. They adapt to resistance, and they just basically constantly draw upon nature's natural intelligence. Of course, they don't have a lot of free will. But imagine if we could live like cells in a tissue, but still have choice and free will to make the right decisions about what we do. It would be pretty good for us and pretty good for the world. And so epigenetics and neuroplasticity, the keys there. I'll end with this poem by Rumi. Motes of dust dancing in the light, that's our dance too. We don't listen inside to hear the music. No matter, the dance of life goes on, and then the joy of the sun is hiding a God. So I'll stop there, and thank you very much for your attention.
Thank you, Rudy. Thank you, Rudy. And he kept on time. No, we can take a couple questions. We'll ask Selen to come up and set up her computer. Just go ahead.
There we go. I would like you to say something more about inflammation. The only test that I'm aware of, the clinical test, is C-reactive protein.
Right.
So what exactly is inflammation in the body?
So this is very different than the inflammation in the body. C-reactive protein and won't get you there. This type of inflammation, I didn't have time to get into it in this talk, but you have in your brain cells called microglial cells. And these are normally acting as housekeepers and cleaning up the brain, watching after neurons. They engulf debris and get rid of it. This is what happens during the slow wave sleep. Well, what happens is these same housekeeping cells, if they see too many neurons dying, they assume that something's going wrong in the brain. And 25,000 years ago, that would have been meningitis or some infection in the jungle. So the microglial cells, when they see too many neurons dying from tangles or plaques, they switch from housekeeping into soldiers, and they start shooting out free radicals, oxygen-based bullets. They shoot out pro-inflammatory cytokines and get the brain worked up, and you get this innate immune response going, because they think the brain's under attack when they see neurons dying. They don't know it's not due to a virus or a bacteria. So what we have to do is, so now we know two genes that control microglial inflammation, CD33 and TREM2, and now we're doing big efforts to try to modulate those genes to stop inflammation. Do we go on? Or it looks like I can answer stuff after, too. So.
Yeah. We're going to have a panel at the end. So let's thank Rudy for a fantastic talk.
Thank you.
This should be over there. So our next speaker, I'm very pleased to introduce Selen Atasoy from the University of Pompeu Fabra in Barcelona, and the University of New South Wales. She's been coming to the conference for a while and had a wonderful paper recently, Nature Communications. And she's going to talk about exploring neural correlates of consciousness with connectome harmonics and neural field models. Welcome, Selen Atasoy.
Well, thank you. Thank you for the introduction and also for the invitation. I'm really honored to be here. Well, I will talk about two concepts, connectome and the harmonics, and I will try to demonstrate how we can use it to explore neural correlates of consciousness. But I want to actually start with an observation. So if we look at nature, we see that all physical phenomena are governed by some kind of physical or natural laws. But when it comes to the brain, we so far haven't yet discovered any fundamental principle or a physical law governing the macro scale, the large-scale brain dynamics. I mean, of course, we know a lot about individual neurons, but so far the complexity of the human brain has defied an intuitive understanding of their collective behavior. So if we look at what we actually do know about the brain, first, a fundamental characteristic of human brain activity, or actually mammalian brain activity, is coherent oscillations. And these oscillations cover a very wide range of frequencies, ranging from 0.05 hertz all the way up to 500 hertz. And we also know that the frequency and amplitude of these oscillations actually change for different states of consciousness. For instance, we know that the daily awake state is mainly dominated by the low amplitude, high frequency alpha oscillations, which then turn into big, large amplitude, low frequency delta waves in deep sleep. Now, what is very interesting about these oscillations is actually they are coherent or synchronized among spatially distributed parts of the cortex. And for instance, you can easily observe that in resting state functional magnetic resonance imaging data. But resting state fMRI has been initially thought to be pure noise. But if you take a seed voxel, like the one which is shown in this video, and compute the correlations between the time course of this voxel and the rest of the voxels lying on the cortex, you would see very, very structured correlation patterns. And when you change the seed location, the structure of these correlation patterns change, but what seems to be always the case is that they are all distributed throughout the cortex. And although these correlation patterns, also called functional connectivity patterns, have been identified in resting state activity, so they form the resting state networks, so-called, these resting state networks surprisingly overlap with the functional networks of the human brain. And these functional networks actually have been identified previously with various different cognitive paradigms. Now, besides these functional networks, there is one other network that has been found consistently, and that is called the default mode network. And what is particular about this network is that its activity actually decreases when you are involved in any cognitive task, and its activity is more when you are not doing anything, when you're resting. And probably because of that, initially this network has been suggested to be a good candidate for the neural correlates of consciousness. But later on, it was a big surprise when Vincent et al. Have actually reported that they have found the equivalent of the default mode network in anesthetized monkeys. And following that, other studies have found the default mode network in humans in light sedation, in general anesthesia, as well as in light sleep. And in fact, in light sleep, there is another study which reports that not only the default mode network, but other resting state networks are also detectable. Now, okay, these findings kind of indicate that the absence and presence of the default mode network or other resting state networks may not be directly the neural correlate of consciousness, But today there is actually significant evidence showing that, that significant changes in anatomical configurations of these networks actually do happen with loss of consciousness. And if you look at these studies, they report firstly that the frontal cortex decouples from the rest of the default mode network in deep sleep and also in anesthesia. And as we also heard yesterday, a recent study by Bartfeld et al. Reports that when we lose consciousness, these functional connectivity patterns, these correlation patterns, seem to be very limited to only a few, whereas the brain in conscious state seems to explore a very wide repertoire of these functional connectivity patterns. Now, this is what we know about the space and time in the brain. So this is what we know about the temporal oscillations and the resting state networks and how they actually change with loss of consciousness. But if you want to kind of solve this puzzle, there are two other equally important pieces. One of them is that we know from neurophysiology that there are two different types of neural activity. So there is excitatory as well as inhibitory neural activity. And we also know that the interplay between these two types of activity actually gives rise to these spatiotemporal dynamics that we measure and we observe in the brain. And furthermore, we also know that loss of consciousness is accompanied either by an increase in inhibition and or a decrease in excitation. And finally, the last piece of this puzzle is the neuroanatomy. So how do we relate these functional and physiological observations to the anatomy, to the particular structure of the human brain? Now, very recently, with the development of the diffusion tensor imaging, we are now for the first time enabled a new technology to actually map the long-distance white matter thalamocortical connections. And as you can hopefully see in this video, these connections actually connect distant parts of the cortex. And the set of all of these connections is termed connectome, or the human connectome. Now, we do know that this structural connectivity, or the human connectome, actually shapes the functional patterns that we observe, the correlation patterns. But we haven't yet discovered a fundamental principle relating structure and function. So the question we asked is, how can we fit all of these pieces of this puzzle together? How can we derive kind of a fundamental principle relating structure, function, and also neurophysiology in loss and recovery of consciousness? Well, at this point, I will actually propose that we do set the brain aside just for a moment, and we look at nature and we look at other natural phenomena and the fundamental principles governing these natural phenomena. So if we do look at nature and various different natural phenomena ranging from fluid dynamics, electromagnetism, heat diffusion, wave propagation, and even quantum mechanics, we actually see that one particular mathematical object, an operator called Laplace operator, or the Laplacian, actually lies at the heart of all of them. And whenever we observe certain patterns in nature, they all seem to be predicted by the so-called eigenfunctions, or characteristic functions of the Laplacian, of this operator. So maybe you can think of the Laplacian as a mathematical object that estimates local anomalies with regard to a point surrounding. So in general, an operator takes a function and maps it into a different function. So it takes a pattern and transforms it into a different pattern. And eigenfunctions are the functions that map back onto themselves. So they are the stable patterns which don't change if you apply the operator, or they only scale. And the eigenfunctions of this particular operator, eigenfunctions of the Laplacian, actually predict electron orbits in quantum mechanics, standing wave patterns emerging in mechanical vibrations, particularly easily observed in acoustics, electromagnetic interactions in a grid of ions, and also besides these physical phenomena, they are surprisingly also, they provide the building blocks of biological pattern formation and morphogenesis. Now I want to quickly actually illustrate these Laplace eigenfunctions or these harmonic functions at the example of mechanical vibrations. So any system that is capable of vibrating has certain natural frequencies, like even this bridge that you see in this video. And if you do manage to excite the system at one of these natural frequencies, something really remarkable happens, and the bridge starts to shake because standing waves form on the bridge. And this phenomenon of standing waves has been actually first illustrated by a physicist and a musician called Ernst Chladni. So Chladni came up with a very, very simple experiment. He took a metal plate, he put some sand on the metal plate, and then he let the plate vibrate. And he observed that in its vibration, actually standing waves form on the metal plate. And the sand starts accumulating at certain regions. Now, this is happening because the standing wave pattern actually divides the metal plate into domains which oscillate with the same frequency but in antiphase. So meaning that certain parts of the plate move up, whereas others move down, and then they switch. And of course, the sand accumulates at the boundaries of these regions. Now, there are only two factors that determine the shape of this standing wave. One of them is the geometry of the metal plate, because the standing waves always adapt to the underlying geometry. And the other one is the frequency of oscillation. So these standing wave patterns only emerge for certain natural frequencies, and when you do change the frequency of oscillation, automatically also the shape of the standing wave pattern changes. And for increasing frequency, you would observe increasingly complex standing wave patterns. Now, although all of these patterns are very, very different from each other and seemingly very complex, they're actually all predicted by one simple equation, and that is the eigen decomposition of the Laplacian. So the eigenfunctions, the size, would give you the shape of the standing wave pattern, and the corresponding eigenvalues, the scalar values, would relate to the frequency of oscillation. So in a way, what this equation does is it kind of relates or decomposes, let's say, space and time. So it relates the spatial pattern of standing wave to the frequency of temporal oscillation. Now, coming back to the brain, the question we asked is, could it be the case that this ubiquitous mathematical framework of Laplace eigenfunctions, of the decomposition of Laplacian, which in this particular example of standing waves actually relates space and time, relates the spatial correlation patterns to the frequency of oscillation, could it be the case that it also applies to neural activity? So could it relate the spatial patterns that we observe to temporal oscillations and to the anatomy of the human brain? Well, actually, to test this hypothesis, we have to apply this famous Laplacian to the particular structure of the human connectome. And this is exactly what we tried to do. So we took the magnetic resonance imaging data and we extracted the surface of the gray matter, so cortical surface of the gray matter. And then we took the diffusion tensor imaging data and we extracted the thalamocortical fibers. And then we combined these two different types of connections, the local gray matter connections and the long-range white matter connections, in a graph structure. Now, the good news is that in graph theory, there is actually an equivalent of the Laplacian defined for graphs, and you can apply it to any type of graph. So using this graph Laplacian, we can actually extend this well-known Laplace operator to the particular structure of the human brain, to the human connectome. And now that we have actually defined the Laplacian on the connectome, we can estimate its eigenfunctions, so the harmonic functions. And we call them connectome harmonics. And if we look at these connectome harmonics, what we see is, first, actually as expected, these harmonic patterns give spatially distributed patterns of correlation. And secondly, also as expected, these correlation patterns or these connectome harmonics, their complexity increases when we increase the frequency of the corresponding temporal frequency. But what we actually didn't expect to see was that one of these connectome harmonics was visually very similar to the default mode network. So we thought maybe this was specific for this particular subject, and we wanted to look at the data for 10 different subjects. And if we do that, we actually observe that consistently there is one particular connectome harmonic that is actually visually matching the default mode network. And if we evaluate this similarity quantitatively, so we used mutual information, so we looked at the mutual information value between the default mode network and first 40 connectome harmonics. And we saw that for one particular connectome harmonic, more or less in the same frequency range across all 10 subjects, we see a significant increase in the similarity, in the mutual information. And also, if we use a different type of measure to look at the predictive power of these connectome harmonics for the default mode network, we again see for the same frequency range a significant increase in the predictive power of that particular connectome harmonic for the default mode network. And if we average across 10 subjects, we still see the significance of this effect here for the same frequency range for both measures. Now, if we look at other resting state networks, first we observe that each of them actually matches significantly a different part of the connectome harmonic spectrum. And secondly, the low-level sensory networks, which you see on the left, actually match better the low-frequency connectome harmonics, whereas the higher-level cognitive networks actually match connectome harmonics distributed across the spectrum. Now, this analysis actually has looked at individual connectome harmonics separately and their similarity and predictive power for various resting state networks. But because different frequency connectome harmonics are orthogonal, meaning that they are independent, actually the set of all of them gives you a kind of a function basis, or you can think of it like a new representation in which you can represent any two-dimensional pattern of neural activity. And in fact, there is one important property of this representation that makes it very, very suitable. Now, remember that these harmonic patterns in general always adapt to the underlying geometry, to the underlying domain. And actually, if you compute these type of harmonics or the Laplace eigenfunctions on a circle or on a string in one-dimensional domain, you would get sine and cosine functions, which are actually nothing other than the very well-known Fourier bases. And in geometry processing, people have extended this idea to other three-dimensional structures, such as a sphere getting spherical harmonics, or any three-dimensional manifold, like the manifold harmonics, and they use this new representation to do some geometry processing because it's way more suitable. And because we define the connectome harmonics exactly the same way as the Laplace eigenfunctions, but this time not on a string or on a circle, but on the human connectome, what we are actually doing is nothing other than extending this Fourier basis to the particular structure of the connectome. And the same way that you can represent any one-dimensional function as a combination of sine and cosines, you can also reconstruct any two-dimensional pattern on the cortex using this set of connectome harmonics. So here you see, for instance, an example of the reconstruction of the default mode network using the connectome harmonic basis. And from bottom to top, we see that our reconstruction actually gets more and more accurate the more connectome harmonics we include. But even for 0.05% of the whole set of connectome harmonics, we actually get a fairly accurate reconstruction. So what does that reconstruction tell us about the frequency content of the resting state networks? Now, if you look at the reconstruction error for different resting state networks, we actually see that the low-level sensory networks can be relatively accurately reconstructed from only a narrow range of low frequencies, and the higher-level cognitive networks actually require a broader range of the connectome harmonics for them to be accurately reconstructed. Now, this is also consistent with the single harmonic analysis that we have looked at earlier. And what does it indicate then? Just to summarize briefly here, connectome harmonics actually provides a connectome-specific extension of the Fourier basis in which you can represent any spatial pattern of neural activity. And in this new language, the oscillatory networks of the human brain actually match harmonic patterns with certain frequencies. Now, these results kind of suggest that maybe this ubiquitous framework of Laplace eigenfunctions of these harmonic patterns actually may provide a fundamental principle relating the temporal oscillations, the resting state networks, and also the human connectome. But the last question that we asked is, if it is really true, if these harmonic patterns really emerge in neural activity, then what would be the neural mechanism creating them? I mean, how do they emerge? Well, one actually candidate mechanism that gives rise to these type of harmonic patterns in dynamical systems is actually self-organization. So self-organization in dynamical systems, let's have a look how it can happen. So first, you need to have two different types of elements, an activator and an inhibitor. For instance, in this picture we see patterns, and in this analogy, the activator would be the little fish, because wherever there's a little fish, other little fish would like to be close by. And the sharks would be the inhibitors, because the little fish would like to keep a distance to the sharks. And the interplay between these two different types of elements can naturally give rise to self-organizing patterns. Now, today there are various different mathematical descriptions for various different self-organizing phenomena, but if we look at the history, we actually see that the first mathematical description for self-organization was actually proposed by Alan Turing as a chemical basis for morphogenesis. Now, Turing suggested that two or more reactant chemicals which diffuse at different rates can naturally give rise to self-organization. And these models, today called reaction diffusion models, have been actually very, very successful in predicting various types of animal coat patterns or shell patterns, and recently even why we have 5 fingers. And there is one very, very remarkable relation between these type of self-organizing phenomena or reaction diffusion type of models and harmonics. And the initial stages of actually pattern formation by reaction diffusion mechanisms poses the same type of mathematical eigenvalue problem as that describing the vibration of thin plates. And we have seen the vibration of thin plates is described by nothing other than the Laplace eigenfunctions. Now, this relation has been shown by Murray and his colleagues, not only mathematically, but also experimentally. And they have done this really, for me, really cool experiment where they cut the metal plates in shape of animal skins, and then they captured the vibration modes. They captured the standing waves emerging on these metal plates. And for different frequencies, they observed different type of patterns, actually very similar to animal coat patterns. But these reaction diffusion type of models are not only good for describing morphogenetic phenomena, but they have been also very successfully applied to predict and describe various types of cortical pattern formation. For instance, they have been used for predicting the ocular dominance patterns in early visual areas, and also the visual hallucinations, various types of visual hallucinations that we can experience. And in fact, the very widely applied neural field models are nothing but an instance of these type of reaction diffusion models. So if we have a look at how self-organization happens in neural field type of models, so in the neural domain, the activator this time would be not the little fish, but the excitatory activity that excites excitatory as well as the inhibitory neural populations. And the inhibitor would be, again, the inhibitory activity, which inhibits both type of populations. Now, if you combine these type of reaction dynamics with a spatial propagation mechanism, something like a diffusion, for instance, you would observe that this coupling naturally gives rise to the self-organization of patterns. And in fact, only by changing the spatial extent of this propagation, only by changing the spatial extent of diffusion, the very same model, the exact same equations would give rise to various different types of patterns. Now, this is exactly how the neural field models actually describe neural activity in the thalamocortical system, but at mesoscopic scale. So what they do is they idealize the cortex like a two-dimensional continuum, like a two-dimensional sheet, so to speak, but the way they model diffusion is they apply a diffusion kernel to this two-dimensional continuum, to the cortex. Now, the question that we asked is, can we then extend these type of mesoscale models to the full structural connectivity of the human brain? So can we extend it from the 2D idealization of the cortex to the full structural connectivity in three-dimensional domain? Well, in order to do that, actually we have to define kind of an equivalent of this spatial propagation, but this time on the human connectome. So we have to model in a way the equivalent of diffusion on the human connectome. And the good news, again, is that now our well-extended Laplace operator actually also lies at the heart of the diffusion process. And now that we have actually defined or applied this Laplace operator on the human connectome, what we can do is we can plug that term in the diffusion, we can numerically simulate diffusion on the human connectome, and then we can integrate that into neural field models. And if we do that, we actually observe that the oscillatory patterns naturally self-organize from the interplay between excitation and inhibition. And very interestingly, if you actually decrease the excitatory parameter in the neural field model or increase the inhibitory parameter, we would observe a slowing down in the cortical oscillations. And this is actually what we know from neurophysiological changes in loss of consciousness, because we know that excitatory activity would decrease or inhibitory activity would increase, which is also accompanied by these low-frequency oscillations, for instance, like the delta waves that we observe in deep sleep. And if we have a closer look at the seed-based correlations in the default mode network, we would also observe that the frontal cortex actually gradually decouples from the default mode network when we increase inhibition and decrease excitation in our model. Now, this is also consistent with what people have reported for loss of consciousness. So they find that the default mode network, the frontal cortex actually decouples from the default mode network in deep sleep and in anesthesia, which is the same type of behavior is observed in these neural field models for decreasing excitation and or increasing inhibition. And finally, if we actually analyze these type of neural field models by the connectome harmonics, because remember, the connectome harmonics are actually the building blocks of the emerging patterns in neural field models. So if we follow the analogy of music, the way I would like to think about it is that the connectome harmonics would correspond to pure notes, and the complex neural field models would correspond to a combination of a few of them. So the way these neural field models create patterns is by letting certain connectome harmonics grow and by forcing all others to die out. So if we look at which connectome harmonics neural field models actually activate for a certain range of parameters, what we see is that if we fix all the other parameters and if we decrease excitation in our neural field model, what happens is that the model becomes very limited to a narrow range of low-frequency connectome harmonics. And the exact opposite is true for inhibition. So if we increase inhibition in this type of neural field models, what happens is that the connectome harmonics that are available for the model to create patterns actually collapse to a very narrow range of low frequencies. Now, this is also in line with with the study of Bartfeld et al., which has reported when we lose consciousness, these functional connectivity patterns are actually very limited to only a few, whereas the brain in the conscious state seems to explore a very wide repertoire of the functional connectivity patterns. Well, in this framework, these functional connectivity patterns would correspond to the connectome harmonics. Now, to summarize, in these neural field models, actually connectome harmonics naturally self-organize from the interplay between neural excitation and inhibition. And in these models, increasing inhibition and decreasing excitation actually yield slower, larger amplitude cortical oscillations, decoupling of the frontal cortex from the default mode network, and also a limited repertoire of functional connectivity patterns or connectome harmonics. And this is what we also observe in loss of consciousness. And finally, because of the ubiquity of this mathematical framework of these Laplace eigenfunctions or the harmonic patterns in nature, because of their emergence in various different natural phenomena, it's kind of very tempting to suppose that maybe the human brain dynamics at the macroscopic scale also follow the same underlying principles as other natural phenomena. I would like to thank all the colleagues and collaborators who have actually contributed at various different stages of implementing this idea, and Human Connectome Project for publishing their great data, and would like to thank you all for your attention.
Thank you, Selen. Let's take a few questions. While Anirban is setting up for the next,
Thank you. That is incredibly exciting work. I can't wait to read the paper. So many implications. So the question I had is that the areas that you're showing at that frequency that you're calling default mode network, to me it looks like it's encompassing all of the medial cortex except for the visual network. So that means that including default mode network, you're also getting the medial components of at least dorsal attention, frontoparietal, and salience. And then on the lateral surface, it looks like also there's such a big swath that it's also containing some portions of some of those other networks. So, but it still seems functionally very significant that those are coming out in the analysis at that certain frequency. So could you say anything more about functionally what you think is going on? Because I think it seems like it's too, you're showing too much cortex to call it just default mode.
No.
But what are your thoughts about that?
Yeah, no, totally. I mean, the one that was best matching the default mode network, actually what we have observed was it was the 9th connectome harmonic, and then 18th and 27th and so on. But because the others are very detailed, like the higher the harmonics, the less likely it is to match any kind of pattern. But we did actually observe for some individuals that it was significantly matching the 9th, 18th, and 27th, and so forth. And of course, the lower the harmonic, the less complex is going to be the pattern. So that's why in the low frequency harmonics, we would observe a broader area that is likely to have more than the default mode network components. But if we would threshold it, you would actually see that it would match the default mode network more accurately. I think it depends. I thresholded it by 0 because that's easier. I mean, it's positive or negative. But if you change that significance level to a bit higher, you would get less and less areas. And I think the higher frequency connectome harmonics are also matching the default mode network. So it's not only one, but it's higher harmonics as well. And the higher the harmonic, the more detailed it's going to be.
Thank you.
So when you use the Laplacian, in the thin plate spline work of Murray, you actually see the biharmonic operator being used and not the Laplacian. So what I wonder is, what would happen if in the graph connectome, where you're essentially discretizing the Laplace-Beltrami operator, what would happen if you use the biharmonic instead of the Laplacian? So it would give you different eigenfunctions. Maybe you can tell us why you didn't do that.
Sorry, if you use what instead of
The
Laplacian?
thin plate that you mentioned, for instance, Murray's work, uses the biharmonic rather than the Laplacian. So it's like the squared of the Laplacian operator, fourth order operator.
Ah, okay, I see. But
So what would happen if you discretize that for the connectome
I
rather
see.
than the graph Laplacian?
I think, so as long as the Laplacian is involved, the eigenfunctions would remain the same. If you have,
The biharmonic eigenfunctions are going to be different from that of the Laplacian. So that was the basic question, actually.
okay. I mean, that would be very interesting to look at, actually. Yeah,
Okay. Thank you.
thank you.
Let's take one more question over there, then we have to move on, and we'll have time for the rest. Sorry about that.
Just quickly, your model basically looked at the spatial connection based on the anatomy, right? But the activation pattern that you observed is based on sort of the temporal propagation of the neuronal activity or bold activation. And that should be propagated through the blood dynamics or synaptic dynamics or something like, that contains temporal component. So I really don't understand why you can actually get something like default mode network-like thing without incorporating how the temporal signal actually signal temporarily propagates, not maybe through the direct axonal connections in connectome.
Actually, the way we think about it is that the dynamics are the ones which are creating these type of harmonic patterns. But the harmonics, because they are the building blocks of these type of nonlinear models, what happens is the interaction between excitation and inhibition, including the spatial propagation, is actually giving rise to these type of harmonics. And I don't think you would ever observe one single harmonic in the brain, because the brain activity is way more complex than just one simple frequency. The same way that we won't observe a perfect sine wave, we wouldn't observe only one of them. But I think the easiest way to think about them is a new way of looking at your data. So we are not saying that the default mode network is emerging just boom as a vibration mode, but what we are saying is that the dynamics are giving rise to these harmonic patterns. So if you split it into these harmonics, some things that are not obvious in other representations may become more and more obvious in this representation. So.
Thank you. Let's give Selen a round of applause. A star is born. Okay, our next speaker from the National Institute of Material Sciences in Tsukuba, Japan, is my good friend Anirban Bandyopadhyay, who will speak on electromagnetic resonance, triplet of triplet fractal. Anirban. Timer, please.
Okay, so there are two kinds of philosophers in the world. One who believes that brain is a computer. So they are the mainstream, and then there are outlaws who believes that brain is not a computer. And when you come to this conference, you very well know who are the outlaws. So I do not need to introduce you. So there are several attempts that are going on all over the world to map the neural network in the brain as accurately as possible. And the majority of the theories and hardcore mathematics simulations, most of these actually belong to those classes of scientists and philosophers who believes that brain is a computer. Those who believe that brain is not a computer, a Turing machine, or the decisions that you are doing cannot be algorithmically or sequential processing, we cannot do it with this. We cannot say what is the alternative, why exactly we can't do it. So our investigations for the last 11 years has been to understand what is the true nature of information in the biological systems and in several natural systems. And you know that in the normal Turing machine or normal computers, we say bits as the information unit. And we strongly believe that whatever be the complexity of the problem, we can write sequence of codes, instructions, and eventually we can generate it. That means if everything is fine, then maybe 200 years or 300 years later, we will be able to regenerate the human consciousness in some computer. So they believe it. So since we do not have any very strong footing like the Turing machine or the Burton-Russell's later of arguments and then sequential algorithms, we know very well that the brain does not, when I came here to speak, I didn't prepare every single sentences. I'm just speaking from my heart. So I didn't write any algorithms. So it is not an algorithmic process, but if it is not an algorithmic process, then what is that process? So it is not, the question is not about getting some equation and fitting it. Today I would say quantum computing, tomorrow I would say another kind of computing, but many of us do not understand that once you get into that bit and other processes, there is no way out. In the last 100 years, there are at least 10 to 15 different kind of formulation scheme who wanted to topple the Turing machine, but all of them failed. And it was shown, whatever be the hypercomputing roots, it is not possible to break the Turing machine paradigm. Nobody could. So this is, so what we do, we show examples that look, there are examples where this person does not have the cortex domain due to hydrocephalus, and still he is doing fine. This is the brain of a Chinese lady at the age of 23. When she went to the doctor, they found that cerebellum does not exist, but she was doing fine. So these are the kind of examples that we show, and we feel good that, look, we have given you a trouble. You are spending billions of dollars to map the brain, and you see that some person who is conscious, who is living very well, he does not have it. So what are you doing? But that is not. But that is not the thing, you know. If we really want to create an alternative world of decision making, we need to give some real concrete science. And without that, we will only make fun of them and they will make fun of us. So this is more of a timeline of the technologies that I used and the results that I will present. In the top 2008, we took microtubule. We started our research with microtubule, but we extended. Everything is not about just microtubule, I would like to say beforehand. So we put the electrode and we did the measurement. Then we put electrode and microtubule on top, then microtubule and then electrode on top. And by 2012, we went on improving our system because we wanted to understand what is the true nature of information. Is it bits? Is it qubits? I published paper with bits, but today I will show you that from 2008, I will take a figure which will be the prime point of my talk. I will start from there and I will demonstrate that if you look at the data long and long enough, you can discover something very vital. So 2016, we have now with antenna-connected atomic probe, very sophisticated. So for 3-4 years, we have worked with an Israeli company, and then we built it. So we can measure. What I wanted to say is that it is a rigorous, year after years process to minimize the noise and carry out experiment by which we can exactly find out how the information is encoded in the system. So this is a plot of resonance. What is actually resonance? Resonance is, suppose you have a flute, and when you blow, there is a cavity, and it interferes, and you get a particular kind of sound, right? So microtubule is basically, it's a non-conducting, it's a damn non-electronic material. But when you trigger it with a particular frequency, it starts conducting. So it becomes interesting. So it comes to alive. But when I plotted this data, and my students plotted it, my postdoc plotted this data in 2008, 2009, we looked at this data and never could figure out there is a triplet of triplet. It was after 4 years we realized there is a triplet of triplet. So I took out the manuscript last year sometime in October and November, and I was looking at this data. And I found that after 2012, I wrote the manuscript to submit. I published some other papers on resonance, but this data I didn't publish. I felt that something is there and I'm missing. If you look at the figure number F, you will find that every single resonance response has associated with a phase. What is phase? Suppose I am asking you a question and you are replying to me, but you have a different mental state with you. So I talk to all of you and you give some response, but some other mental state with you. So when you interact with each other, you state something that is a response that we measure. But at the same time, you have some other mental state which influence other kind of operations. For an example, say two waves come together, say superpose, and after that, if they have a phase relationship within them, then you will find A plus B plus something. This is how we differentiate classical with quantum. So there was a phase which was very important, and phase relationship. So philosophically, what I figured out is that, and these resonance frequencies, they change among themselves. Vibrations change among themselves like a music. How is it happening? So when we are trained to practice science, we are said that you pump a system or ask a question to a system, and with a probe, you get an answer. Then you get many different kind of answers or frequencies or responses, and you correlate them, feed it with the equation, and then you give a proposal. That is how scientific practice is all about. So this is what we have been doing all these years. Until recently, when things went very different. So what we found is when you are getting the response, when you are hearing the bing, the information is not there. Information is between two bings when the system is silent. So nature is encoding information not when it makes a sound or makes an expression, but when it remains silence. And between many, many different kind of silence points, if you integrate them together, you can see a beautiful pattern and beautiful information. So this is fundamentally very different what we have believed in the last 100 years. So it is not the response, it is the silence. So for the rest of my presentation, I would like to show it that what kind of silence is this and what could we make out. So these two videos will run very fast. You may get. So earlier we had a concept of beat and qubit. So two classical points, and then you have a phase. Now, I suggest that you take time cycle as the unit of information, because you can find it anywhere. And if there is a small singularity, then where you get the sound ting, that is the classical point. Now, using time cycle, you can connect many, many time cycles together, and you can present many different forms of information. So there would be not much problem. I assure you, we have written several tons of codes to do that. So when you see the resonance peak at the top and at the bottom in the crest, these are the classical points which you can see. And in between, there is a flow, which is phase, which you do not see. But microtubule-like system, they have large number of resonance frequencies, and all of the frequencies have particular phase. So they are alive. When you integrate them together, you need to put them in a different plane and project in the hyperspace. So what you do, earlier you need qubit as information. Now you had only 2 poles, North Pole and South Pole. Now you have many, many poles on the sphere, on the Earth planet. And those poles are interconnected. And those time cycles, which actually holds the information with Bing and gap between the Bings, project in that sphere on the spherical surface, and you get the information. So I know that you have not understood it very well. And I also understand that I tried to understand it many times, and I even couldn't. So what we did is we wrote a program, and we tried to understand that if nature encodes information, then how to do that? So basically, a system, when it is in the singularity mode, that means I can't see. My body is just like a singularity. I mean, it's opaque. I can enter inside. Say this is a network, and time cycles. Just you think that this is a path, you can walk along these paths, okay? So you will have, when you are looking at it, you have your mental state, and when you are looking at particular object or a particular person, then the world inside it has a mental state, and the world outside it has a mental state. So these 3 mental states together define the information. So what happens, it is just like those who know that Turing tape. You can write any instructions line by line on a Turing tape. Every single cell has a Turing tape inside. So this single statement is sufficient enough to break down the Turing paradigm. So when we outlaws, we will try to fight with them. With the Turing machine, we need to come out with a simple mathematical statement. And I say that every single cell has a Turing tape inside. You just mathematically apply. Everywhere you will find singularity. Everywhere there is a singularity. So what I will do is we will take some example, and we will try to process some information. So what happens is, in the conventional science, when we say bits or qubits, then you need a software code to write those instructions. But now we have a time cycle which synchronizes with the external system, and it gets information by itself. How? Suppose you have a circle, and you are increasing the diameter of the circle. So you move towards high frequency to low frequency, because the path you have to cover much longer, right? This you have understood, right? Okay. The next thing, you need to understand there's 3 things, and we can do it. So if these relative diameters of 2 circles goes decreasing, then you need to multiply with a higher number, so the intensity increases. Okay? So the second thing that you need to know. The third thing that you need to know is, in a time cycle, how phase is encoded. So these are the 3 points, and you want to write a triangle. What you do is you see the length of the perimeter changes, right? So you will not be interested in where the points are. You will be interested how long is the path. So in the triangle, you saw that you change the points, and thus you can do. So what happens in a circle, the information is not in the circle itself, but some circles inside. So you can go inside and inside and inside and inside to find where is the information, and you can never find it. So where is the information? Information is on the path. That is the gap between the 2 singularities, gap between the Bing that you get. So that is the philosophy that I wanted to demonstrate. But if you see it, then what happens is when you look at many, many dots and try to connect with the software that normal scientific practices we do, we end up always into a wrong result. So I will show you with a frog that if you try to do information, if a microtubule or the tubulin or any biological system wants to see information, then how do you encode in reality? So this frog, I will try to create time cycles for every part of it, and triangle, for triangle we put another time cycle. For every single geometric shape, I put one, another, something, and then I rotate the time cycles, and then we will be able to generate how the vibration would be projected on a sphere. So these are the results which we derived from the experiment. So this is the frog, how it looks like. So if a microtubule sees it, or a protein sees it, it would like to see like this. Anyway, so I avoided some set of experimental results, and I represent it with this because this is the simplest way I could explain how we do encode information, okay, in the phase space, which we cannot see. So when I'm talking to you, you are giving some response due to politeness and many other reasons, okay? But your actual mental state is never expressed. Those are expressed between the silence, between the responses, interactions between other two. Those we need to extract. So the same data, we need to read it in a very different way. So now I would like to get into some of the experiments and try to demonstrate you that why we come to this kind of conclusion, okay? So in the textbook, you have studied neuron firing, and this is a particular neural spike. What we did is we imaged that when the neural spike is going on, what is happening inside, okay, at a higher frequency level. So we believe that brain is just like a cavity, and we have a cavity inside, and cavity inside, and cavity inside, and cavity inside. And all are connected by slower to faster and faster and faster and faster clocks. So we found it experimentally. So we found that when the neuron is firing, actually, there is a faster clock inside from the microtubules, which is keeping the time. So when neuron is firing for 1 millisecond, someone else is processing information at microsecond. Then in the proteins we go, and proteins we find that proteins are keeping time at nanosecond scale. We have gone to picosecond and even faster scale, and even in the slower scale. So there is a clock inside a clock, inside a clock, inside a clock, inside a clock. So these are kind of time resolution videos. So both the things, the middle one is the ionic transmission that is going on, and the right one is the dielectric transmission that is going on. So earlier we did not have the technology to look. So your textbooks teaches you that you have membrane, and that is ionic transmission goes on, and that is everything about biology. All the multi-billion-dollar projects that are going on for brain building all over the world, they believe that information is being processed through the membrane. Nothing happens inside. So I'm not saying that everything happens at the microtubule level. I'm saying this is one fraction of many, many, many clocks over there. So there are several this kind of presentations. So I would like to show you the live imaging of microtubule clocks. And if you look at this, then you would see that when it is resonantly vibrating, the lattice of the tubulin proteins on the microtubule surface is changing. So it's beautiful transmission. So this world was completely unseen from the scientists. So we believe that membrane is everything over there, but such a beautiful things are happening. You are pumping with a classical signal, and all the tubulin proteins that are arranged in a lattice, they are changing, and this is a quantum mechanical description. So there is a connection between the classical and the quantum process, okay? And microtubule you cannot image at a very low temperature. it will break apart. You need to get to a higher temperature to get this kind of image. So we did not stop at the microtubule. We went inside, and we looked at what tubulin protein does. So tubulin protein also dances with the music, and it keeps the time at a much faster clock, okay? And you will see that not only tubulin protein, we went even inside the tubulin protein at the secondary structure levels. And even then, we found the clocks are operating, okay? So, and this is monomer. Monomer has 8 different kind of cavities with there. But I would like to tell you that we do not need to think that microtubule is the only thing that is doing this. There are several other proteins which we studied, and all of them has this clocking behavior. And luckily, my postdocs and students are so great. I told them that, is this the cavity thing? Is the only thing? They studied 26,000 proteins. Can I believe it? 26,000 proteins from the PDB database. They brought it down, did the simulation, and found that only 17 kinds of different cavities are there. Only 17 kinds of different clocks are there, which is repeated. Okay, and the most complex clocks are in the biological, in the blood proteins, and tubulin is in the middle. Okay, it is not very simple. It is not very complex in the middle. So these are some of the examples of proteins, and there are plenty of other proteins which we have studied. What we get is a connection of clocks, but how those clocks are connected. Okay, when we talk about clock, it is very easy to talk about phase. You know, from phase we can come to clock because the time between the 2 tick is important, and that comes to us very naturally. Okay, so this is an experiment which is very different philosophically than the conventional resonance measurement that we do. So in this case, what we have done is normally we used to ask questions. Suppose I have a device, I ask a question and I get an answer. Okay. Now what we have done, we ask the same question from different side. So if somebody believes in silence, then you can probe him from different direction and you will get different answer. Philosophically, this is true for humans, and this is true for even the biological systems. So when you look at these data, try to feel that a silent system is being asked questions from different directions, and they together evolving a pattern which is invisible in the normal way of asking questions. So in the neuron is there, or microtubule is there in the B, and the tubulin proteins are there in C. But we are asking questions from perpendicularly from different directions. And it is giving a triplet of triplet. Triplet means 3, and pair of 3, and that also pairs into 3. But the beauty of the system is that we are pumping the frequencies from 2 different directions. Those phases are getting overlapped, and they are triggering the nerve spike. Okay? So the nerve spike that we see has a world inside. So this is a other group's data. The sound that you are hearing is the spike of the nerve spike or the neuron spike. So we are able, with our technology now, to look inside one tick. So this is microtubule and tubulin band structure. And we would dissolve just one peak. So you see the triplet of triplet we will cover. So all this music are played. When you listen to this music, try to understand between two sounds there is a phase relationship. That I have also converted into sound so that you can feel how actually the information is encoded. All these things happen within a microsecond. And several such music is played, or the activities take place, which eventually results into a single spike. So what I want to tell you that what we see with our eyes, these sounds, if I do not add the phase, then you will be tick, 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 very fast. Not this one, right? So try to feel it with a phase relationship. I'm sorry. This is not for entertainment. So, so when we, so when we understood that, there is a clock inside a clock inside a clock inside a clock, and then we can't cut the brain into different parts and then try to understand. So one thing that disturbed me a lot, that is, why are we getting triplet of triplet? Why nature selects this kind of thing? So we went on and did some very simple calculation, which you learn at the age of 8 or 9. So that calculation is here. So we take a simple cavity, just like a flute, and you do LCM and HCM, you learned as a kid. Many of you have forgotten. But if you remember, then you can take, say, 12, and then if you look at B, then you can see that there are 12 loops that makes 12. 2 into 6, 2 loops, each contains 6, that can make 12. Then 3 into 4 can make 12. 4 into 3 can make 12. 6 into 2 can make 12. So these are the harmonics. These are the nested cycles are there. And then you combine them at the order factor. The mathematical formulations or mathematical derivation is quite complex. So it's not that thing. But if you plot these integers with the nested cycles, you will be able to see beautiful patterns. And those patterns make into a fractal, triplet of triplet fractal. So it is coming from the natural number system. So in the number system, so we did some very interesting plot. If you look at A, it is the how many numbers are there in the universe which are divided by 2? Can anybody tell me? How many numbers are there in the universe which are divided by 2? 50%, right? Because every alternate numbers you go, it is divided by 2. And how many numbers are there which are divided by 3? So you have to divide 100 by 3 because every third number is divided by 3. So 33. But there are some of these. So there are numbers which are divided by 5, 7, 11, 13. So in this way, you can make a combination. And if you see 66% cases, everywhere there is a fractal structure in nature. From this plot, you can figure out you'd always get a triplet of triplet band. So the 3 numbers will occur always there. Because you can't create a new pattern. So you need only a few primes, and then you can cover 99% of the space. Another very interesting thing we got in B. What we did, we put the natural numbers, and we wanted to know how many oscillators do we need. Because microtubule, if we consider tubulin as an oscillator, and microtubule, then microtubules combine together to make the action core of a neuron, and then many neurons come together to form a cortical column, and then cortical columns come together. So ultimately, eventually, the whole brain is built up. So how many oscillators would you like to have to reach a saturation? Then we need to make a unit, and then start. Suppose you have a small balls. How many small balls you would like to put together to make a bigger ball, and those bigger balls will come together? So how many that number? When we plotted with this, we found that the triplet, the teardrop-like shape. You see, this is a teardrop-like shape. This teardrop becomes a sphere or ellipsoid at the number 10 to the power 11 or 10 to the power 12. And you will find this number particularly everywhere in nature. Okay? I can give very popular example, but I do not want to entertain you. You find out yourself. Okay, so what we did is, just go ahead. So mathematically, we try to create the structure. So one mathematical equations, and you will find this kind of structural evolution almost everywhere if you start scanning from the atomic scale to the largest to the whole brain. Okay? And so this is the, so if you look at a sperm is just like the first structure. The whole brain with the spinal cord is the first structure. And ovum is ellipsoid. And when sperm goes to ovum, a new life is born. So you will see this composition of structures everywhere. So what we did is we went on analyzing the dynamics and a brief overview of the whole system. So what we did, again, I asked the students to go through every single structure that we have in the brain and survey the literatures. And we went on cavity inside a cavity inside a cavity with every single component. And we found their structures, whatever available, whatever nearly available, and then we correlated with the simple 7 structures, teardrop to ellipsoid or sphere. Only 7 structures were the only 1 equations, and we could easily go. So if we increase this pattern, if we go on inside the first layer, so this is just an overview. It will take, you saw the connectome result earlier. So connectome is just like combination of 8 or 10 cavities. The third layer, there are 12 different layers. So I would not like to go into details. I'll just switch off there. So what we are suggesting is that in the current biology, what we are said is the vertically you see a blue line. So that is the frequency range where ionic nerve impulses and other frequencies are over there in that region. In our research, we tried to study the electromagnetic frequencies. That is also a small region. What I'm suggesting is the whole frequency wheel is the total biological system of our human brain. And if we want to understand, we need to understand the whole wheel. So what the existing biology has studied or emphasized is a fraction. What we have studied is also a fraction. So we can make small frequency wheel and complex and more complex systems with nested rhythms. We can continue. And finally, so this is the website where we are uploading the fractal geometries or the cavity geometries of every single cavity that human brain has. Even microtubule or tubulin, and even inside the tubulin, the cavities that you have, even the DNAs or the other cavities. All the CAD files will be there. So you can 3D print with a $300 machine in your room, house, and then you can find out how music is being played inside the system. We are doing it in our laboratory. It's fantastic. So this global database of biological rhythms and 3D printing, and then 26,000 proteins that we have studied with the cavity resonator, fractal cavity structures, that we are also going to upload in this website. So in conclusion, silence is important, not the being. That's philosophically that what we learned. So different kind of silences are there between different beings. So the gap between the singularity is important. Debate is not about classical or quantum. We need nested time cycle-based concrete mathematics to explore singularity to find more and more silence or peace. Okay? So we need to explore peace in every single being that we hear. We are developing a global database of biological rhythm, a free cavity printing file database to print human brain and a protein cavity-based database for finding a common frequency region for musiceuticals. Musiceuticals means music-based treatment of things. Here are the students, some of the students who worked with us, and thank you.
Thank you, Anirban. I'm not sure we got all that, but we'll sleep on it for a few years. And let's have Rudy and Selen come up. We have time for a little, for some discussion. In the meantime, any specific questions for Anirban?
Question for Anirban. Anirban, last year at the TSC, you were only talking about the hierarchy of cycles. And now you seem to have given an interpretation on how information is deeply hidden. What is your next step? How do you find the source of information?
Every year I think that I have understood information, and that after a few months I realized that it was a mistake. What can I do? Last year I felt that I was confident that it is the time, but I did not. The problem is that talking big is easy, and then implementing it with program and verifying it. For example, I can say that information is processed in the biological world with this protocol. Then you get back to your own programming, then simulate information, regenerate, encode information, and then decode it, the thing that I showed. So if all those hard steps you need to go through, and then you have to talk. That's a problem. So last year I did not figure out the phase part. So I nested up to nested time cycle I went. But the phase part was missing. So now after phase part, we have now successfully processed information with those protocols.
Thank you. I think you're going somewhere profound, but maybe you have written your book too soon?
Yeah, actually, 2010. Since 2010, I'm writing the book. For others, it's very easy. I told you. Every time I write the book, it's now 10th version. I deleted it completely because I think everything is bullshit after writing the book. And then because I don't know what is information in the biological system. Many people are very happy without knowing and telling everything. But when I don't know what is information in biology, what I am talking about consciousness, what I am talking about brain, right? So if you do not know information, you know nothing.
Yes.
So first you establish what is information.
Yes. Thank you so much.
We'll have to share this mic. Andrew.
Thanks. It was a great plenary. So this is a question for Dr. Atasoy. So I'm interested in the implications of your theory for brain dynamics, and I wonder, is it too far to say that your connectome harmonics could be considered some sort of eigenstates of a Hamiltonian, a sort of brain Hamiltonian? And if so, I wonder, could you perhaps generate an evolution operator from that? And you said you want an underlying principle of how the brain works. Is it possible to come up with something like a Schrödinger equation based on your harmonics?
Yeah, it could be considered as a simplified version of that. We are actually starting a collaboration with some other labs now which have different equations similar to self-organizing ones that I showed, but different where you get actually damped standing waves as the solutions. And the stable solutions of that are harmonics as well. And I think that relates to the Hamiltonian, because in Schrödinger's equation you have the Laplacian and you have an extra term to that, which results, as far as I know, in damped type of equations. But we are going to look into that. It's tempting, but I think it's a bit early to claim that yet, but I'll keep you posted.
Thanks.
Hi, this is for Anirban. I was unclear about when you were talking about singularity and what the role was, and whether that was an artifact of the reference system you used, or that was something you deliberately induced, or that was something you found naturally. You know, there's like half a dozen interpretations, but you breezed through that so quickly, I didn't follow that.
Yeah. It's a record system that we use. It's the observer's singularity. Every single singularity is observer singularity, because if you have higher resolution, then you go inside and you find some other information. Today I didn't show the quantum interference pattern of microtubule, but it's beautiful. If you look at it, any part you zoom and redo the experiment, you find a new interference pattern inside. So it never ends. So that's kind of a, so it's the resolution of the observer that defines the singularity.
Okay.
Hi, I apologize that I'm wearing pajamas. I actually have a serious question. This is more directed towards Rudy. I read a paper recently that stated that it's a well-observed phenomenon that in the late stages of Alzheimer's, when the person is in like a vegetative state, and their brain, under brain scans, it undergoes like mass destruction of brain tissue. In about 8% of people, they undergo like a period of terminal lucidity within about the last few days, where they get perfect, their cognitive function returns, and they do weird things like they call up all their relatives and loved ones and tell them they love them and give them a final farewell. And I like to think, and then they pass away a few days later. And I like to think that it's maybe an explosion of release of neurotransmitters that causes that. Do you have any theories of your own?
I think terminal lucidity is real. I mean, there are, I didn't know about the 8% number, but certainly a small fraction of patients, and maybe more if we asked about it. Yeah, for some reason in the last day before they go, after being even in a complete vegetative state, suddenly recognize their loved ones and come to and say goodbye. And no one's really explored it. No one really knows the basis for that. But it's worth exploring. I agree. It's a pretty amazing phenomenon. In terms of a theory, I mean, you'd have to be, you could say you're activating synapses suddenly, but if those synapses have been destroyed, that's not a good answer. So it's a mystery.
So maybe it supports Deepak's hypothesis.
Microtubules. Stuart says microtubules. So. I'll go with that.
My question has sort of ties in what I think it's directed towards Selen, but tying it with what Anirban is talking about in terms of scale-free nature of the frequencies 1/f, and if the way that you have in your model, I don't actually remember what you showed about like how the space affects time. The oscillations, but if it affects actually the whole spectrum of oscillation, the frequencies, the scale-free parameters or something like that.
You mean towards the end of the presentation, the relationship to the model, or?
Well, by manipulating parameters of your model, does it actually change the pattern of frequency response across frequencies? Okay. Because that's been shown to correlate with behavior, like 1/f slope has been shown to correlate with behavior in a lot of tasks.
Yeah, what we observed for the neural field model was, it's a very complex nonlinear model, so you can't see everything that it does, but what you can do, and what we did, was to fix all the parameters and look at one at a time. And in that case, when we actually decreased excitation, we saw that the red plots that I showed were theoretical analysis called linear stability analysis. So you just look at which of those patterns can be activated. But then we actually numerically simulated the emerging patterns for the same parameter range, and that actually confirmed what we observed theoretically. So it seems to collapse to a narrow range of low frequencies when we decrease excitation or increase inhibition, which is also what we would expect from neurophysiology.
Okay, but you haven't looked at the relationship between different frequencies?
The relationship between?
Between different frequencies, nesting and stuff like that. We can maybe talk about it later.
Sure.
Good presentation by all of you. I had a question for Rudy. And as you mentioned in your presentation that the, can you hear me? Can you hear me up there?
No, a little closer, please.
You mentioned that the mouse is a notoriously bad model for Alzheimer's. And you used the mouse example saying if you can reestablish the connectivity between the neurons, the memories are still there. Does that still apply to humans where you have a lot of neuronal death? That's my question.
Yeah, I had a little trouble hearing you toward the end on the question itself.
With the rodent models, the neurons tend to remain alive, but you lose
so the information is being stored, but the synapses on the spines of the processes are being pruned back.
Right, so my
And
question
that's
is...
probably the reason why you can't access them, right?
Right, but with humans, where you actually have death of neurons, is that still going to apply, or do you think the memories are truly gone? Does the mouse model apply to humans?
Yeah, I mean, it's always tough to, I mean, most of the time, mouse models don't apply very well. So I think we can't just jump. I mean, when it comes to Alzheimer's, anything you use to treat a mouse just about works. I mean, it's crazy. And then they do trials, and it doesn't work so well. But I think in this case, you know, given how basic what they're doing is, you know, it's optogenetics, it's a trained behavior, they're looking at memory. The mouse with the neurodegeneration from Alzheimer's is not remembering, and it's because those neurons are not getting stimulated. The spines are not there for them to be activated, and then doing that artificially, optogenetics, suddenly the mouse remembers the task. My bet would be, given that we also see these spines being lost on dendrites of neurons in the molecular layer of the hippocampus, that I'm guessing that it is going to correlate from mouse to human, but we'll have to see. Yeah. I just want to make a comment, too, that on your default network pathway that you resolved, it's almost the exact same trace of how tangles spread through the brain. And during deepest sleep, you know, default mode networks turned off. And this is the time when Deepak would say you reach into your soul because you don't have all your personality in the way. But it's also the only time you don't make amyloid in the brain. And amyloid is inducing tangles. So I think there's a real clue there about maybe even more than we thought before, importance of default mode network in terms of how the pathology is forming and spreading. So it's an interesting juxtaposition, you know. Yeah. Yeah.
Have you tried Subrata's radiofrequency vibrations in your gel system?
Right, so Stuart asked if we tried radio frequency. Subrata sent us an excellent postdoc. Anirban sent us an excellent postdoc, Subrata Ghosh. And we're using radio frequency stimulation to try to tear apart amyloid. And we've used it, we started simple and liquid dishes, and now we've gone to the 3D cultures. I can just say, you know, the results are encouraging enough that we're planning more studies, and we are finding frequencies that seem to help rid the brain of amyloid. What's the frequency, Kenneth?
The frequency that you're using to treat the plaques.
Well, we can.
Oh, that's proprietary.
Well, you know, the thing is, I look at it this way. I would share anything at any time, but when it's another person's career who's younger involved, I have to protect them. So, okay. Yeah. So.
Did you have a response to this question?
Well, yeah, actually, it's a very it's very nice to know about that because we just started looking into sleep fMRI data using this method as well. And what we find is that the connectome harmonic that is matching the default mode network seems not to be most active in deep sleep. So that kind of also confirms that the Alzheimer's part, but I mean, it certainly has potential to look into. I'd be excited.
Speaking of frequency, I've spent the last 20 years using audible frequency, bouncing it off the body and the field that surrounds the body and listening to the signals that come back. And I've discovered that it appears that memory may be stored in the biomagnetic field that surrounds the body. And this makes sense in the context of if microtubules acted as antenna to receive and transmit the information contained in these standing waves. If the microtubules begin to degenerate, then the ability to access that is inhibited. However, the memories would still be stored. Given that laws of physics state that magnetic fields guide and influence electric currents and activities, I'm just wondering if any of you have considered the biofield or the biomagnetic field and its role in generating these frequencies, harmonics in the brain, and storing memories.
Yeah, you can you can search in Google, Sahu and Bandyopadhyay, and you will get our patent, US patent we got last year, that describes how we can store charge as a function of magnetic flux. So all the technical details are there. Yes, magnetic flux is produced, and we are now trying out, in our laboratory, if you come now, we have, at least 30 or 40 different versions of analog devices of microtubule created by graphene and other materials, where we produce regularly magnetic flux. And in my iPhone, full of these apps where you can map the magnetic flux. So I carry it with me always, so we can easily check how the magnetic flux is being changed. So magnetic flux does play a fundamental role, and it helps. It has several interesting things. I mean, you just go through those. I cannot detail here, but if you go through the patent, you can find all details of this magnetic flux production.
Thank you.